Thank you, everybody, for joining the last presentation today. And the topic's going to be precision coupling for determining antibody titers and screening antibody specificity. Now, while that's the topic of the presentation, we're actually going to cover a little more. We're going to share information on how you can do various things that can be used for these kinds of applications, but are also applicable to developing a lot of other Luminex-based XMAP bead studies. So what we're going to cover today is a variety of topics, which include, if I can get this forward, okay, let's see here, this thing doesn't want to forward. Having a technical difficulty I thought we worked out already. Let me get rid of the thing there. There we go. Okay. So the topics we're going to cover today are going to be a quick review of the coupling chemistry that makes the platform so versatile. We're also going to go over some of the resources and tools that you can use for developing a variety of different assays on the bead system. And then we're going to focus on some of the techniques that you can use to couple either at molar amounts or microgram amounts of capture molecules, mostly proteins, for developing different assays. We're also going to look at some of the tools that will allow you to do the calculations for doing these different types of couplings, because these can be somewhat complicated procedures to perform, and using these tools can help you do the calculations and perform the protocols pretty easily without making errors, as if you were going to do it with, say, a hand calculator in a notebook or a notepad. Then we're also going to look at tools for making your multiplex bead mixes, because while making a good Luminex assay is dependent on how you couple your capture molecules to the beads, developing that assay and having it perform reliably is also important to make sure you're making your bead mixes properly. And then we're going to have a short summary at the end to summarize everything. So the coupling chemistry. What makes the Luminex platform so versatile is the carboxyl groups that are on the bead surface. Because when you use EDC to activate these carboxyl groups, and in the case of coupling proteins, you'll add a sulfonyl NHS to stabilize that very reactive ester. Once that carboxyl group is activated and stabilized when needed, when you add an amine-containing compound, such as a protein or maybe an amine-modified amino acid, that amine molecule will be covalently linked to the beads and be on there stably for a very long time. So the types of molecules you can couple include purified proteins or antigens. Antibodies are commonly coupled. You can couple peptides as well, although there are sometimes some tricks you need to do to get these coupled reliably onto the beads. Some carbohydrates that have been modified to have amine groups on them can be coupled. You can also couple a number of small different molecules, such as some small molecule linkers, some drugs, and some hormones. And if you want to develop a genomic assay, you can couple amine-modified oligos to the beads as well. So some other coupling modifications that can be done are things such as coupling protein linkers. And a number of protein linkers that can be coupled and have been used in the past include BSA, chelimpet hemocyanin, thyroglobulin, and avidin. There's been a lot of people that have used avidins for biotin tag molecules, and we actually sell avidin beads. So if you're interested in doing something like this, you can contact your business manager, and he'll help you determine, what you need to do in avidin binding, so you don't have to couple these yourself. But for some other applications, there are some small molecule linkers that you might want to couple. For example, ADH. This is a good coupler or linker that can be used for coupling peptides to the beads. There's also MBPH that can be coupled to the beads as well, with a linker that gets it off the surface or closer to the surface. And that's used for a lot of sulfidol-containing peptides or proteins. And you can also couple biotin to the beads if you have things that can bind biotin that you want to bind to the beads as well. Now, getting back to protein coupling, when coupling proteins, large proteins such as immunoglobulins are pretty easy to couple because they contain a lot of lysine residues that have an primary amine group that's way off the surface of the protein, which makes coupling these in multiple orientations pretty easy to do. Some smaller proteins, such as various smaller antigens you might want to couple to the bead, they can be usually coupled fairly effectively as well, depending on their lysine content. When you start getting into peptides, Some peptides might not have any lysines in them, providing that amine needed to couple to the activated carboxyl group, or some might have just a few. So when coupling peptides, I recommend that you contact your FAS to help guide you through the process of what's the best method to get that peptide coupled to your bead. So no matter what type of assay you want to develop, we have a number of resources and tools to help you develop your assay. The best tool is the XMAP cookbook. The 5th edition is out now, and the cookbook essentially is a collection of easy to follow protocols that has lists of reagents and equipment that you would need to build just about any assay. So while these protocols are generally good starting points, we still recommend that you contact your FAS to go over what your particular assay needs and to design with you the best protocol and the best modifications to existing protocols to help you build the most reliable assay. So, as I mentioned earlier, the carboxyl group is the key to getting anything coupled to the beads. And in the case of the carbodiamide coupling, the step of coupling or activating and coupling these molecules to the beads is pretty straightforward. And for something like coupling proteins directly, the process takes about 3 to 4 hours. And it all starts with taking your beads out of the stock vial, washing them, putting them into the reactive vial, which is a non-binding tube, washing them with water or activation buffer, then resuspending them to a certain volume in activation buffer, then adding sulfur NHS and EDC in the case of doing protein couplings, and then incubating for 20 minutes to activate all those carboxyl groups on the bead surface. After that 20 minutes, you'll want to wash the beads with activation buffer or mesh buffer, and then you'll add your protein, usually diluted in PBS or activation or mesh buffer, so that you're adding a certain amount of protein per million beads. And then you're going to incubate that for about 2 hours. Now, the best way to do this incubation is on a rotary shaker that can kind of rotate and agitate the beads for that 2-hour period, as opposed to sitting at the bench and vortexing your tubes every 10 to 15 minutes. So if you have a rotator, you can better spend that 2 hours doing something else more productive than sitting at the bench and vortexing every 10 to 15 minutes. And if you need recommendations on the rotator, we can provide that if you go this route. And once you've done your coupling reaction to the activated beads, then you'll wash out all the residual protein that might not have bound and any residual EDC or sulfur NHS that might still be there. And then you'll resuspend and block the beads in a PBS TBN buffer. Now, there are some situations where the PBS TBN buffer might not be optimum for you. And again, that's why you should engage your FASs to talk about what you want to do with the beads and what you want to couple, so we can determine if this is going to be the best washing and blocking agent for your particular assay. So let's talk now about some of the advantages of coupling at molar values versus microgram values. The cookbook, for example, in its basic coupling protocol, suggests using 5 million beads that you activate with the EDC sulfur NHS, and you couple that with 5 micrograms of protein. Now that translates into 5 micrograms of protein per million beads. And this is another concept we want you to keep in mind. Whenever doing these reactions, you should keep in mind how many micrograms or moles of protein are you reacting per million beads, because it's that stoichiometry you want to keep consistent when you're starting out doing these initial assay development experiments at low coupling scales. But then later on, when you have to scale it up, you want to keep some of these stoichiometric ratios the same to hopefully get the same type of reactivity in the beads when you scale up compared to the small scale tests you've done. So, but users can play with this ratio. There are some situations where you might need less than 5 micrograms of protein per million beads. And we generally recommend when testing an assay and developing it, that you try 3 or 4 different coating levels, with the 5 micrograms being at the high end, and then going lower, probably sometimes as low as a microgram or even a half a microgram of protein per million beads. And we recommend doing that testing to figure out which coating level is going to give you the best performing assay. But this kind of coupling is generally suitable for many applications, especially coupling antibodies and beads for sandwich capture assays. Now, for some small proteins and peptides or protein linkers, you can still use this microgram coupling per million beads. And again, you're going to want to do some titration to figure out which coding level is going to be the best for you. In some situations, however, coupling your proteins at moles of protein per million beads is better when it's advantageous to have the same number of molecules on the bead surface with when you have molecules of different molecular weights that are going into the bead mix. What this does is that by having the same number of molecules on the bead surface for all the different molecules in a mix that have different molecular weights, it allows the signal, the MFI intensity, to be specific for the specificity of the target that you're looking at on the bead and not dependent on the number of molecules on the bead. So this type of approach is really good for a variety of serological assays that can benefit from the molar couple, which we're going to talk about next. Because way back, I was working on a project where I had a 10-plex assay that consisted of 6 antigen-coded beads and 4 control beads. And the purpose of the assay was to detect antibody levels for the 6 pathogen-specific antigens. And the serum that we used was from animals that had been infected with different amounts of pathogens and different combinations of pathogens. So the 6 antigen-coded beads consisted of proteins of these different molecular weights. Now, they're not significantly different in their molecular weights compared to other studies that we've done, which I'll show you in a minute, but they were significant enough that some of these had cross-reactivity. So serums that should have been very specific for one of these also showed some high specificity for some of the other ones. So initially, when we started doing this study, we started doing the couplings all at the microgram coding levels per million beads. And we were seeing that, we still weren't getting some of the responses that were supposed to be specific for one antigen to be as highly specific as we thought they should be. So we started testing some picomole coding levels and comparing them to the microgram coding levels. So an example of that is this data here, where we tested coupling these 6 antigens at either 5 microgram per million beads, 2.5 micrograms per million, or 1.25 micrograms per million. And the scale over here is in fold over background. And the background in this case was an IgG strip serum. And what we can see is this protein here that's about 36.25 thousand Daltons is the one the serum should be reactive with. And the level of reactivity at 5 micrograms is pretty good. It's around almost 500-fold, but some of the lower ones are almost at 100-fold. Then the 2.5 micrograms is at about 1,000-fold over the background. And the 1.25 is just below 800-fold over the background. Not much change in the relative amount of the other background beads, showing some cross-reactivity. So what we did is we compared coupling these same proteins at 3 different molar ratios of either 20 picomoles of protein per million beads, 10 picomoles, or 1 picomole. Now, some of the backgrounds cross-reactivity we saw was still present on the other beads with this one serum, but the fold specific signal for the antigen was much higher relative to what the backgrounds that we saw on the other antigens. So much so that we're now talking about 20 picomoles, a specific signal of about 3,000. At the 10 picomoles, we're seeing a specific signal of about 3,500. And at a lower picomole coding, we're seeing a signal to noise ratio of somewhere on the order of almost 5 to 6,000. So by using these picomole coding levels, we're able to more fine tune and increase the specificity for the antigen that the serum was supposed to be responsive to relative to the non-specific cross-reactivity we saw on the other 5 beads. So some of the advantages of doing molar coding versus the microgram coding is, first of all, with the microgram coding, you're going to use a lot more protein and it's going to be more expensive. So for example, when we were coding those 6 antigens at either 5 micrograms, 2.5 or 1.25 micrograms versus coupling at 20, 10, or 1 picomole, the actual microgram amount that we had to use with the molar coding was significantly less than doing 5, 2.5, and 1.25 micrograms. So for example, here you can see that the equivalent in the microgram per million beads coding levels was all below 1 microgram per million beads for each of those 6 antigens at all 3 of the picomole coding levels. So this is going to significantly save you a lot of money and expense in coding your beads at the picomole coding level compared to doing the standard 5, 2.5, or whatever microgram coding that you would normally do for this type of assay. Now, while for this assay, coupling at equal molar amounts seem to work fairly well, there are some situations where you might not want to do the coupling at equal molar amounts. For things like screening antibodies that are being generated, if you're doing recombinant antibodies and you want to see how specific a recombinant antibody is for its particular antigen versus other antigens, doing the equal molar amount, again, is a good option because all the molecules will be at the same, all beads will have the same number of molecules, and the signal is going to be based on specificity, not the amount of molecules on a bead. But there are some situations where you might not want the equal molar amounts. And here's an example of that. This was in relation to a COVID study we did with a collaborator up at the University of Rochester, where we were using spike protein, which had a molecular weight of 135,000. And we also had the receptor binding domain, which had a molecular weight of about 30,000 Daltons, which is about 4.5 times smaller than the spike. We also had a nucleocapsid fusion protein of the N-terminus and C-terminus subunits. The fusion protein had a molecular weight of about 46,000, which is almost 3 times smaller than the spike protein. So the assay consisted of 5-plex, which was 3 of the beads had the antigens of spike, receptor binding domain, or nucleocapsid on them, coupled at either 10 picomoles or 100 picomoles per million beads. And then there were 2 other control beads in the mix as well, one to monitor the addition of the anti-human IgG and one to monitor instrument performance. Now, the reason why we didn't do the equal molar coupling is because spike was a much larger protein and had many more epitope binding sites and antibody binding sites to give more signal than the 2 smaller proteins would. So what we wanted was we wanted the assay to preferably be positive for all 3 of the antigens, as opposed to only being positive for one at different stages of infection. So in order to bring the signal up for the RBD and the nucleocapsid to hopefully be positive for a true infection, we coupled those 2 smaller proteins at a higher copy number of molecules per bead. And as we see here, this sort of worked to some extent when we're looking at PCR positive samples with days from symptom onset from 5 or less up to 21 days. And we could see that compared to an Abbott Architect system, which we were comparing the assay against in that lab, that the Abbott Architect only used nucleocapsid, and this one was using all 3. And we were already, be able to get more correct positive calls in these PCR positive samples at these different time points than the Abbott Architect was at early stages of infection. And as the infection progressed, the positive rate for all 3 antigens started to coincide more with each other, suggesting that we probably could have tweaked the coding level of these 2 smaller antigens a little more to have it become more correct positive calls early on in infection. But this was the rationale for doing the 2 different coding levels of the smaller coding level for the spike versus the higher coding level for the RBD and nucleocapsid. So in order to do these kinds of couplings, trying to do these calculations with a notepad and a calculator can lead to a lot of problems. So we have a variety of tools that you can use for doing these calculations, which makes it easier to do it reproducibly and more error-free. And these are generally a couple of Excel workbooks that can be used for doing your microgram couplings of proteins or peptides or whatever you want to couple to the beads in microgram quantities from small to large scale couplings. And as I mentioned earlier, we make avidin beads, and there's a workbook for doing the microgram per million bead coupling for avidin beads as well. We also have a workbook for doing the molar coupling of proteins at different scales, and the same thing for doing molar bindings of avidin beads with your biotin tagged proteins or other molecules at different scales as well. So the way these workbooks work, and here's an example, is everything generally you'll see on these is where the green fields, you always put in your text or numerical data to do calculations. The blue fields give you data that you need to do and to monitor for certain things like what volume of beads are you going to use, how many bottles you're going to use, what does it mean with regards to the molar concentration and things like that. And then down here in the bottom part, this is all going to be the activation step up here. Down here, when you pick your molar coating levels you want, it will actually tell you how many molecules per bead you're actually coating when you're doing the molar coatings. Now, this also is a little more complicated than doing microgram coatings, because since you might be coupling different proteins with different molecular weights, where the stocks also have different milligram per ml concentrations, there's a tab here that helps you do all the calculations to get all those different proteins with their different molecular weights and different concentrations to the same molar concentration. So when you're then doing coupling with all those at the same coating levels, you use the same volume to assemble all those reactions with, which eliminates a lot of errors if you don't do those kinds of adjustments. So again, we have the capability of doing those calculations if you're going to be using the avidin coated beads for coating your molecules at molar levels with the avidin beads as well. And then for the microgram coating, it's a little more straightforward because you don't have to compensate for molecular weight. All you need to do is get all your proteins to the same milligram per ml concentration, and you're good to go with doing the coupling reactions at virtually any scale. And the same is true for the workbook that does the same calculations for doing the binding of biotin tagged molecules to the avidin beads. While doing these couplings and doing them precisely is important for building your assay, it doesn't end there. To increase the precision of your assay's performance, you also need to make these bead mixes more precisely. And we have a number of tools to do that as well, because again, doing these sometimes with a notepad and a calculator leads to mistakes. But by doing the workbooks here, you can actually get all your beads after you've coupled, all the beads that have to go into a single multiplex mix. What you want to do is you want to get them all to the same concentration. And generally, that concentration that I recommend is at least 500,000 or higher, depending on the scale of the coupling you did. Because if you go by what the cookbook says of resuspending all your beads to 50 beads per microliter, that translates into 50,000 beads per ml. And right off the bat, if all of your beads are at 50,000 beads per ml, and you start combining them all together in a multiplex mix to put, say, 50 microliters into a well at our preferred concentration of 2,500 of each bead region per well, if you start out with that value, you're actually going to be putting in far fewer beads per 50 microliters than you think, because that level of concentration causes dilution of the beads as you start combining them. So what I recommend after you've done a coupling reaction or several reactions is use this workbook, either this first tab here or the second tab, to figure out how to get all your beads to one concentration of about 500,000 or higher. And then once you've done that, you're going to want to use this next file, which is the multiplex bead mix calculator, to determine how to make your multiplex mix so that when you're combining all of your beads together, when they're all at the same stock concentration, no matter how many plex you have, this will tell you whether or not you need to do a concentration step, which is what frequently happens if you're using beads at the 50,000 beads per ml. So to ensure that you're building your assay properly, you want to use these two tools to properly make your bead mix after you've gone through all the trouble of precisely coupling your proteins or other molecules to the beads themselves. So in summary, no matter what type of assay you're building, the key to building an assay with precision is the following. Number one, if coupling molecules, you need to think of the beads themselves as another reactive component and use them at some, microgram per ml or moles of protein per ml concentration when doing the couplings. And if you download this presentation in a PDF format, this here is also a link to some of the spreadsheets and information on how to do this. The next thing to do is after you've done your coupling or binding to avidin beads, you always want to determine the bead concentration afterwards. Because one thing you want to do is you want to make sure, let's say if you've done 5 million beads in a coupling reaction, you want to make sure you're getting back most of that 5 million beads or close to it, so that you can determine, bead loss or things like that, and the effectiveness of your coupling protocol. And then you also want to then, dilute your beads all to the same concentration so that when they all go into the same mix, you can use that bead mix calculator. So you're just using one volume to put all the beads in. Because I've known people that have been doing things like a 62 plex. And, they complain that I've got to change my pipetter 62 times because they're all a different concentration. Or recently I was working with another pharmaceutical company that had a 23 plex. And when I went to train them, their technicians were complaining, now we've got to change our pipetter 23 times to make a multiplex bead mix. How can I do it easier where I don't have to do that? And so taking this step after you've coupled your beads and normalizing everything to one concentration is much easier and prevents a lot of mistakes downstream. So that means that you also need that multiplex bead mix calculator, which you can get through this link here. And so overall, the key takeaway is not only can, for some applications, the molar coupling work very well, but you should, look at these various workbooks and learn how to use them, or contact your FAS and have them walk you through the process to build your assays more precisely. So that is all for today for my presentation. So I look forward to answering your questions in the question answer session. Thank you very much.